Hello children, welcome to AIMS India's online classes, biology session, cell division and cell cycle, assessment 2. This video includes level 2 questions. So, let us proceed. Cell plate grows from, cell plate grows from. So, where this cell plate actually it appears in the metaphase, the cell plate is formed. The cell plate is usually formed in the plant cells. In animal cells, it is called equatorial plate. Okay. How the cell plate is formed or grows from? One cell, one wall of to another, center to wall, wall to center simultaneously. So, actually the cell plate is formed center to wall. Okay. The process in which cells become specialized to perform specific functions is called differentiation, cell cycle, cytokinesis, mitosis. It is the differentiation. Okay. Actually, growth involves cell division, cell maturation, cell differentiation. Cell differentiation is very important. So, during cell division, means for example, the fertilization, first gametogenesis, formation of gametes, then fertilization, fusion of gametes. The fusion of gametes result or the fertilization process results in the formation of zygote, a diploid cell. So, this zygote will undergo cell division, means before formation of gametes one cell division occur. What is that? Meiosis. After meiosis the gametes are formed, then fertilization process takes place. After fertilization a diploid zygote is formed. That zygote develops into an embryo by means of cell division, that is mitosis, cell division. So, after the development or during the development, at some stage the embryo cells are assigned with specific function, or this process takes place. The process in which the cells become specialized to perform specific function, this process is called differentiation, cell differentiation. So, after the differentiation tissues are formed, specialized tissues. Every tissue performs its own function. Okay. So, after cell differentiation during the embryonic stage, ectoderm, endoderm, endoderm and mesoderm, these three embryonic layers are formed. From these three layers different organs and organ systems are developed. Okay, for example, ectoderm, from that skin, nervous system, like that from every embryonic layer some different systems developed. Okay. So, that is organogenesis. First gametogenesis, fertilization, then cell division, differentiation, then organogenesis. This is the development or embryology, which is not the phase in interphase. G1, G2 synthesis, cytokinesis. So, cytokinesis is not the phase. So, between G1, G2 synthetic phase, S phase occur. During which phase do cells suspend? During which phase do cells spend most of their lives, most of their time? Mitosis, interphase, anaphase, prophase. Okay. In interphase, the maximum time, because cell prepares for the cell division, so it will take much time during interphase. Next, which of the following is the shortest phase of mitosis, in which The chromosome line up in the middle of the cell and the spindle fibers connect to the centromere. Prophase, metaphase, telophase, anaphase. So, it is the metaphase, during which phase does the nuclear membrane disappears, centrosomes disappears and the spindle apparatus forms. Okay. Interphase, prophase, metaphase, anaphase. So, during prophase all the cell structures disappear except the nucleus remaining, all cell structures start disappearing, nuclear membrane, centrosomes. Okay. Prophase, during which phase do sister chromatids separate and move to opposite poles? Prophase, metaphase, anaphase, telophase. It is the anaphase during which the chromosome splitting of chromosomes takes place and they move to the opposite poles. During which phase do chromosomes uncoil, nuclear envelope forms and spindle fibers disappears. It is the end of nuclear division. The prophase, metaphase, anaphase, telophase. It is during the telophase. Okay. Next, during which phase does the cytoplasm divide into two identical cells? Mitosis, metaphase, interphase, cytokinesis. It is during two identical cells, means first nuclear division complete, then cytoplasm divide is mentioned here. So, cytokinesis. It is the cytokinesis. In plant cells, what is the structure that forms in between the divided nuclei? We discussed this also. Cell membrane, cell plate, cell wall, cleavage furrow. It is the cell plate. It is formed by the Golgi apparatus. Okay, cell plate is synthesized by during cell division, okay, during metaphase. A cell that has 68 chromosomes undergo mitosis. How many chromosomes does each of the two daughter cells have? Okay, a cell that has 68 chromosomes, okay, it undergo mitosis. How many chromosomes does each of the two daughter cells have? So, here what happened before cell division or before mitosis, interphase occurs. So, during interphase this 68 becomes 136, okay, doubling of chromosomes takes place. So, here 68 plus 68 is equal to 136. So, the two daughter cells are formed, this 68 again divided by 2. So, each cell receives 68 chromosomes. During which phase is DNA replicated or copied? Interphase, prophase, telophase, metaphase. So, it is during the interphase DNA replication takes place. Identify the phase of mitosis. Prophase, metaphase, anaphase, telophase. Here the daughter chromosomes reach the opposite poles. So, here it is anaphase. Next, which is the correct order? A, B, C, D, E, F are given. So, you have to place it in correct order. Okay, let us number. So, this is the first interphase. So, D is the 1, then yes, A is the second, and the next one is F is the third one. Here the figure is somewhat blurred. Next, the fourth one is metaphase, equatorial plate is formed. Then the fifth one, Anaphase. Yes, splitting of chromosomes takes place. So next, the last one is telophase, so that is B. So D, A, F, C, E, B. Okay, so B is the right option. So like this, if the question is given with the images and order the images, means first you can number it and then you can segregate the number in a correct order. Okay. Next, which part? So here the figure is not in the above figure only, means in the previous figure only, which represent the metaphase in the above figure. Here in this figure, which represent the metaphase. Okay. So the metaphase is represented by this C. Okay, children. So here the answer is C. Next, in what stage the chromosomes separated and moved to opposite poles? In what phase the chromosomes are separated and moved to opposite poles? Separated and moved to opposite poles. Start moving is Anaphase, and separated and moved to opposite poles means telophase. Here prophase, metaphase, anaphase and telophase are given. So it is the telophase. Cytokinesis usually occurs after mitosis, means after nuclear division. So if a cell completed mitosis but not cytokinesis, what would be the result? A cell with one nucleus, a cell with one really large nucleus, a cell with two nuclei? None of these. Okay, usually by the end of telophase, two daughter nuclei are formed. So a cell with two nuclei. The point on the chromosome where the two chromatids are held together is, it is the centromere. Okay, here centrioles, okay, they are present in the centrosomes. Centrioles reach the opposite poles and the spindle is formed. The centromere is the structure which chromatids are attached. Okay. Next, the structure of coiled DNA and proteins that form in the cell's nucleus prior to mitosis is centromere, centriole, chromosome, chromatids. So here the structure of coiled DNA and proteins, it is nothing but the chromosome. Chromosomes are formed during cell division. The division of the cytoplasm following mitosis results into two identical copies of the organelle, cell organelle called daughter cells. Cytoplasm, cytokinesis, telophase, mitosis. It is the cytokinesis after which the two daughter cells are formed. A jelly-like substance found inside a cell surrounding its organelle is. So already in the first unit itself you have studied. So cytokinesis, cytoplasm, mitosis, epithelial. It is the cytoplasm, jelly-like substance, in which the cell organelles float. The first stage of the cell cycle, during which the cell matures, grows and prepares to divide and copies its DNA, the cell stays in the most. So entire story explains about the interphase. Okay, it is the interphase during which the doubling of chromosomes or the copies of DNA takes place. Cell stays for long time and prepares for the cell division. Contains the genetic material and serves as the control center of the cell. What is that? Nuclear membrane, spindle fiber, nucleus and telophase. It is the nucleus which is called the control center of the cell. Last phase of the mitosis, in which the spindle fibers break down, chromosomes unwind, and the nuclear membrane begins to form around each of the two sets of chromosomes or two sets of the chromatids. What is that stage? Prophase, metaphase, anaphase, telophase. It is the telophase, last phase of the nuclear division, during which these changes occur. Which is the correct order going from smallest to biggest? Cell, tissue, organ system, cell, organ, tissue, organ system, organ, tissue, cell, organ system. So it is the first one which represent the ascending order, smaller to bigger. Cell to form the tissue, combines to form the organs, combine to form the organ systems. Which of the following is not a part of interphase? G2, M, G1, S. It is a M phase which is not the part of interphase. M means mitotic phase. Okay, remaining all interphase is equal to G1 plus S plus G2. Which of the following statements is true of cytokinesis? Takes place in the plant cells only, completes the cell cycle, organizes DNA, occurs in prophase. Okay, it completes the cell cycle. Cell cycle will ends by the cytokinesis. Which of the following is not a reason we need mitosis? Growth, recovering from injuries, replacement of dead cells, sexual reproduction. It is the sexual reproduction which does not require mitosis. Meiosis is required for sexual reproduction for the formation of gametes. DNA is replicated during dash of cell cycle, G1, S, G2, G0. It is the S synthesis phase where the DNA is replicated. In which stage of cell cycle is the cell preparing for cell division? It is the G2 phase. Finally, the cell prepares for the cell division. Next, where does DNA synthesis happens in a eukaryotic cells? In eukaryotic cells, cytoplasm, mitochondria, nucleus, Golgi apparatus. It is the nucleus involves in the cell division where the DNA synthesis happens. Which of the following terms means cytoplasmic division? Interphase, mitosis, cytokinesis, cell cycle. Cytokinesis. Okay. Next, a key difference between daughter cells resulting from mitosis and meiosis is. So after meiosis the cells are diploid, after mitosis cells are haploid, after meiosis cells are haploid, after mitosis cells are diploid. After meiosis there are 4 daughter cells, after mitosis is 1 daughter cell, after meiosis there are 2 daughter cells, after mitosis there are 4 daughter cells. Okay, actually after meiosis the cells are haploid, after mitosis the cells are diploid. In humans, the 4 daughter cells of meiosis contain 46 chromosomes, 23 chromosomes, 92 chromosomes, no chromosomes. In humans, 4 daughter cells of meiosis contain haploid. Okay. So 23 chromosomes. 23 pairs means 46. In most animals, a cell will contain the following number of chromosomes after mitosis: N, 2N, 4N, 8N. So always diploid daughter cells of mitosis. So 2N. N represent monoploid, diploid, tetraploid, octoploid. Okay, so here diploid means twin. Which of the following stages contains no trace of nuclear membrane? Prophase 1, telophase 2, anaphase 1 and prophase 2. So it is the anaphase 1 where no trace of nuclear membrane. Okay. Next, the spindle fibers attaches to which of the following? Nuclear membrane, cell membrane, centromere, chromatin. Here the spindle fibers are attached to the centromere. So during the metaphase itself, and during anaphase, that centromere is pulled by the spindle fibers, the length of the spindle fibers decrease, so that splitting of chromosomes takes place. Okay. So spindle fibers attached to the centromere. The haploid cells are seen after meiosis 1, after metaphase of mitosis, after telophase of mitosis, before anaphase 1 of meiosis. So here after meiosis 1 or by the end of meiosis 1, 2 haploid cells are formed. Chromosome number is reduced. Reduction division, meiosis 1 is called the reduction division. The homologous chromosome pairs, that is tetrads, exist during which phase? Prophase 1 of meiosis, prophase 2 of meiosis, prophase of mitosis, all prophase stages in the mitosis and meiosis, homologous chromosome pair. It is during the prophase 1 of meiosis. Okay. During the interphase, the DNA is called organelle, DNA, chromatin, cytokinesis. The DNA is called chromatin material during interphase. Okay. What phase is this? Here one image is given. This represents prophase, metaphase, anaphase, telophase. So it is clearly representing 2 daughter nuclei are formed. Okay. So it clearly represents the telophase. Which phase comes next? So already here one phase is given. After that, which phase come? So this phase representing the anaphase, the prophase, metaphase, anaphase, telophase. So here the question, which phase comes next? It is the telophase. Okay, the last question, what is the role of meiosis? Formation of gametes, bringing haplophase, bringing diplophase, completing life cycle. Okay. Role of meiosis, formation of gametes. Okay. Okay, children, I hope you followed me. So read the book, clarify your doubts with your subject teacher. Thank you for watching our video. Please subscribe our channel and press the bell icon for receiving latest updates.